Definition of transcription. Transcription is the process by which genetic information contained in DNA is transcribed, copied into messenger ribonucleic acid, also known as mRNA. It takes place in cell nucleus. Enzyme involved is RNA polymerase. Segment of DNA is known as a gene. Okay. Gene consists of promoter, mRNA coding sequence, and terminator. Promoter specifies where transcription is to begin. mRNA coding sequence contains coding information for polypeptide chain specified by gene. Third, terminator. Terminator specifies the end of mRNA transcript. There are three stages of transcription. First, initiation. Second, elongation. And third, or lastly, termination.
Okay, this is the nucleus, and then this is the nuclear envelope. Okay, DNA. This is the structure of DNA. A DNA section called a cistron contains the genetic information needed to make a particular polypeptide, but DNA cannot leave the nucleus and so cannot be used directly for protein synthesis. This is the pre-mRNA. And then this mRNA. This mRNA template passes out of the nucleus and moves into the cytoplasm and bind with ribosome where it is read or translate into the polypeptide. DNA undergoes the process of transcription from DNA to pre-mRNA. And the pre-mRNA undergoes RNA processes to be mRNA.
There are three stages in the transcription of DNA to RNA. First, initiation. In prokaryotes, occur with specific recognition of the promoter sequence by RNA polymerase. In eukaryotes, more complex RNA polymerase recognize a complex of proteins that help binding of enzyme to promoter sequence. Begin with unwinding of DNA strands by RNA polymerase of promoter sites. The enzyme RNA polymerase catalyzes the process 5' to 3' direction. This is the direction of transcription, DNA to RNA. Only one enzyme involved in transcription. It is RNA polymerase. This is RNA polymerase. This is gene, or we call it as cistron. There are two sites. It is promoter site and terminator site. RNA polymerase attaches to the starting point of the gene cistron, the promoter site initiating transcription. As we know, promoter is a short segment of DNA in front of the gene for the binding of RNA polymerase. Okay, this is the point of attachment. RNA polymerase. At the point of attachment, the DNA helix unwinds and unzips. As we know, during transcription, the RNA polymerase moves along the gene in the direction 3' to 5'. This is the direction. Okay. Next, elongation. Nucleotides are added in the 5' to 3' to the growing RNA strand according to the same base pairing rules as for DNA, except uracil are added instead of thymine because RNA doesn't have thymine. Lastly, termination occur when RNA polymerase encounters the terminator sequence on DNA template. After transcription complete, RNA transcript is released from DNA template.
The next stage is elongation. As RNA polymerase move along the 3' until 5' DNA template, free nucleotides are added in the 5' and 3' directions, complementary to the DNA template. During elongation, nucleotides are added to the growing RNA strand according to the same base pairing rules as for DNA. Except that uracil is added instead of thymine when adenine is encountered on the DNA template. RNA does not have thymine. The last stage of transcriptions is termination. Termination of transcriptions occurs when RNA polymerase encounters the terminator sequence on DNA template. When transcriptions is complete, the RNA transcript is released from the DNA template, followed by the RNA polymerase.
In summary, transcription is the process of synthesizing single-stranded mRNA using specific segments of the DNA as template. There are 3 stages in the transcription of DNA to RNA. First, initiation, followed by elongation, and lastly, termination. The enzyme RNA polymerase catalyzes the process in the 5' to 3' direction. In the mRNA strand, the base uracil replaces thymine. The RNA transcript leaves the nucleus to be translated. The RNA of eukaryotes must be processed before translation. Okay, that's all from us. Thank you for listening.